Hi guys, Miss Francis here today to discuss the applications of DNA technology within the medicine and the pharmaceutical industry, within the environmental industry, as well as the agricultural industry. So modern biotechnology is making enormous contributions in both the diagnosis of diseases and then the treatment of diseases. So for example, PCR is one of those applicable DNA technologies where PCR can amplify small amounts of DNA and it can help detect otherwise elusive infections such as HIV. Medical scientists can also use DNA technology to identify individuals with genetic diseases before the onset of symptoms, even before birth. So it's possible to identify symptomless carriers for diseases such as hemophilia, cystic fibrosis, muscular dystrophy. Techniques for gene manipulation holds great potential for treating disease via gene therapy, where you can change the DNA code that's altered or mutated to the correct code. And this involves using a technology called CRISPR, which has really made some strides in recent years. We're going to do a whole another video talking about CRISPR and the ethical implications of using such a DNA technology. The pharmaceutical industry is using novel ways of fighting diseases that don't respond to traditional drug treatments, such as creating human insulin and human growth factors in bacteria, for example. We have created genetically engineered vaccines. We have also created genetically engineered proteins. So these genetically engineered proteins can either block or mimic surface receptors on cell membranes. So for example, one experimental drug treatment used a receptor protein that causes HIV to bind to it instead of to the blood cell. Therefore, the HIV binds to that drug, the one that's mimicking the receptor instead of the actual receptor itself. So now the HIV doesn't enter the blood cell. Traditional vaccines usually tackle one virus, but scientists have genetically engineered vaccines that can be made to fight various viruses at once. We talked about the applications as far as forensics goes, using DNA fingerprints to identify criminals and violent crimes, to settle paternity suits, to identify the remains of individuals who died during natural or man-made disasters. There are also applications of DNA technology in the environmental vector, where there are microbes that can clean up highly toxic waste. They've been genetically altered to do so. They can also participate in sewage treatment. For many years, scientists have been using DNA technology to also improve agricultural productivity. For example, DNA technology is now routinely used to make vaccines and growth hormones for farm animals. Scientists have created transgenic organisms. Another word for transgenic organisms are genetically modified organisms, or GMOs. You've probably heard a lot of that in the news. But these transgenic organisms have been developed to exploit the attributes of the new gene. So, for example, they have faster growth or larger muscles. There are examples of the antifreeze gene from salmon being inserted into tomatoes to prevent the tomatoes from freezing during transit or while being grown on the vine. In our next video, we're going to discuss how scientists developed a transgenic organism, how they cloned an organism. I'm sure you've all heard of Dolly the sheep. Well, we're going to learn how Dolly the sheep was actually created.